Okay, so today I'm going to talk about work that we just upload to bioRxiv, and it's about the structure and organization of a full-length epidermal growth factor receptor in extracellular vesicles by cryo-electron tomography. Um, so Okay, let me see if this works. Yeah. Um, just a brief introduction for the receptor tyrosine kinases. They are key regulators of the cellular processes, proliferation, differentiation, cell survival, and metabolism, as well as cell migration, cell cycle control. So they are type 1 cell surface proteins, and they all share the same architecture with an extracellular region that will bind to a ligand, a transmembrane domain, and an intracellular region that will have the kinase domain. In humans, we have 20 different subfamilies of these receptors, and today I'm going to focus on the EGFR family that has 4 homologs. So these homologs, they are all implicated in many cancers when they are dysregulated or when they have mutations. So they are described here, and there are already a lot of FDA-approved drugs directed against these different parts of the receptors. Unfortunately, most of the times after the treatment is instaurated, the mutations appear and make resistance to the treatments. So usually the treatments are combined, but even then there's the need to get better treatments. So for these receptors, the canonical mechanism of activation would be when they are in an inactive tether conformation. When the ligand interacts with the ectodomain, they extend the ectodomain, allowing the dimerization of the receptor to happen, and that would activate the intracellular region through a kinase dimer that would phosphorylate the C-tail that would recruit adapters and effectors. So the structures of these receptors have been solved by different like X-ray structures, cryo-EM structures, usually just from the ectodomain purified, the intracellular region purified, but the only time that we were able to get a near a full-length structure was like in 2021 with this approach that they use actually 4 different type of membrane mimetic environments, and that was like detergent micelles, lipid nanodiscs, amphipoles, and peptidiscs. And the result they got is that they were able to resolve the ectodomain, but never the transmembrane or the intracellular region. And same results were described with the insulin receptor. So there are a lot of questions that are still needed to be answered in regards of the full-length protein, especially how it's associated in the membranes. Are really clusters or the oligomers are needed because the receptor is found like in inactive dimers, and it also get dimeric when it gets activated. So our goal in this project was to get EGFR in a more native membrane instead of using detergents. And for that, we tried 2 different approaches. One was using BLPs, making virus-like particles with the receptor on the membrane. And the other one was using an escort cellular machinery to induce extracellular vesicles. So I'm going to try to explain both very quickly. Well, someone before talked about VLPs, so we all know that with the murine leukemia virus GAG protein, you can induce the formation of BLPs without the need to have any like viral pathogenic protein in the vesicle. These VLPs have been used for many purposes, especially like to display antigens on their surface. So we thought, well, if we could display our receptors and the vesicles, the VLPs are small enough, maybe we can image them by cryo-electron tomography. So we design a system that, so our main interest is to have the full length receptor. So in order to have it with its transmembrane domain and the intracellular region, we needed some sort of a puller to get it into the VLP. So we designed sort of a puller that would have like a transmembrane region of a viral protein that would interact with the gag and would be part of the vesicle. And in the C-terminus, we add a streptavidin. So if we added a strep tag in our EGFR, then that would be pulled into the vesicle. And that is what we did. We also test different other pullers. So we could use different type of tags, and those were anti-HA, SCFV, and anti-flag. And the flow through to get to the VLPs was to co-transfect in 3 different plasmids. One that would have like the gag protein, another one would have the puller or the streptavidin, and in a third plasmid, the pulley. So after we got to the ideal ratio of transfection, that would be expressing HEC293 T cells, then we would collect from the media all those VLPs that got released. And then after a few steps of concentration and only ultracentrifuging for sort of purify those vesicles, we would image them by cryo-EM. Of course, we check that the vesicles had everything that they need to have. So that was the receptor we were expressing, both in the lysid of the cells and the supernatant, that was where the vesicles would be, and also the puller. We also checked that the receptor was still active in those vesicles by adding the ligand and ATP to the vesicles, and we could see phosphorylation. So they were successfully expressed on the vesicles and were active. Now, we still had on the vesicle the puller protein, so we needed to cleave those proteins, and we realized at that time that the cleavage was not 100% efficient, and the vesicles would need further purification in order to have a successful imaging. So we tried to move forward and test the second method, that was making extracellular vesicles by fusing into the C-terminus of our receptor, the full-length receptor, a sequence. This is a method that was described by Hoffman et al. In 2023, and it's a sequence that codes for escort and Alex binding motif. So it would recruit escort proteins that would make your receptor get into the membrane that is gonna bud from your cell. So in this case, well, of course, we test that adding that sequence in the C-terminus where the kinase domain is located, the receptor is still active. So it's still being able to get phosphorylated normally and activates a downstream signaling as usual. And we usually perform these assays in a different cell line. So the flow through in this option of getting the vesicles, it's easier because you only transfect with one single plasmid that has your receptor, and then the cells are collected. And because we knew the vesicles need to be purified, we added a strep tag in the N-terminus of our EGFR, and now we add a step of affinity purification with the streptactin column Then we concentrate our vesicles and we perform the cryo-ET. So here I'm showing the results of the purification with the elution fractions. This is a Western blot, blotting against the receptor. We did like a Kumasi gel to make sure the receptor was purified. This is the starting material, and this is after the purification. And by cryo-EM, we were able to see the vesicles with proteins on the surface that had the size and the shape that indicate that EGFR was there, successfully expressed. So the vesicles we got had a range of diameter that it's graphic here. The mean is around 80 nanometers. So we decided to analyze this sample by mass spectrometry, and the majority protein in the vesicle was EGFR. We also analyzed what other proteins were coming together in those vesicles, with the surprise that all the effectors and adapter proteins described for EGFR pathway were present in those vesicles. So that for us was a realization that this method is very powerful to study interactomics. Now, I also analyzed these vesicles were active in vitro, so the receptor was active. And moreover, we also test the method of adding the escort sequence into other transmembrane proteins that had more than one pass into the transmembrane. And here I'm showing 3 examples. The insulin receptor is a constitutive dimer, and it also went very well into the vesicles, and the mass spectrometry showed it as the most abundant protein, the same as a 13-pass transmembrane protein, that is the sodium iodide symporter, and a 7-pass transmembrane protein that was the smoothen receptor. Now going back to EGFR, our cryo-electron tomography help us solve some details of the receptor in regards of how is it disposed on the membrane. So we saw it's the presence of clusters on the membrane of the vesicles. We also were able to distinguish the kinase domain as an intracellular density, and there was a gap between the membrane and the intracellular density that hasn't been described before. So these tomograms were made in the presence of the ligand. So what we were expecting is that all these receptors were in the active conformation. So to have an idea, this is the crystal structure of the ectodomain, and this typical heart shape is what we would expect when the dimer is present. In red, we can see the ligand, and this is around the size we were expecting. So in our tomograms, we were able to overimpose the image of the crystal structure, and we clearly could see all the different ectodomains. Now in the inside, there's like this mud pie. And very interestingly, when we were able to see isolated dimers, we could even see the kinase domain. So we went ahead and we make average of these particles. The ectodomain, we did subtomogram average of 843 particles, and we got a 15 Armstrong map of the ectodomain. And only with those very isolated dimers, we could find only 16 in these few tomograms, and we could have like a rough reconstruction of the kinase dimer. So that was very exciting. We're trying to push that, getting more particles And also within the distribution of the clusters, we were able to describe that they are not in a very order form as it was described before. And this is just a small video of how the tomograms look. And this is a reconstruction using the ISONET to compensate for the missing wedge. So with that, I would like to make my acknowledgements to our lab, is the Dr. Leahy lab. Our collaborators in this work has been the Sulonki lab, Edward Marcotte lab, from Jason McLellan, and from our Department of Structural Biology, Axel Brillat. If you have any questions.
That was a great talk. Thank you so much. I just want to maybe ask Rajesh to unmute. Could you ask the question yourself? It's in the chat space as well. He's asking, can you comment on the yield and expression of target multi-TM proteins using the Hoffman fusion sequence?
Okay, let me see. Yes, so actually the multipass transmembrane proteins were done with the escort domain. So that is the Hoffman method. That was even surprising for them because in their system, they attached the sequence right after the transmembrane. So we attach it after the C-terminus of one single transmembrane domain, but then with all those multipass transmembranes, and it did work perfectly. So I don't know if that answers the question. And then the structure, it says if it was seen without ligands, is different with, we try different kind of ligands. We only did EGF. What we did do is with and without ligand. The thing is that without ligand, our ectodomain is fairly small for cryo-ET. But yes, we can see a clear difference because these more monomeric forms. We add the ligand on purpose to have the very easy to identify heart shape form. I don't know if there's any other question.
Michael, can I have another question?
Yes.
Sure, please.
Yeah, hi, Monica. That was my second question. So, in the Pamela Bjorkman's group paper, they mentioned that the fusion works better on the C terminus. I don't know whether they tried on the N terminus of multi-TM proteins. So can you comment if it would work or it needs to be on the C?
It does work. We actually did it in both ends.
Okay, great. Yeah, I mean, we have been using that since past couple of years, and it's worked really well on single TMs. So it'll be great to push that further.
Yes, yes. Yeah, that's why we try one that had multiple transmembranes, and it did work perfectly.
Thank you.
Great, Scott. You've got a question too. Do you want to ask it? Otherwise, maybe Monica.
Yeah, sure. I mean, there are some membrane-associated proteins, G proteins, for example, that are predisposed to precipitate and kind of unstable. But if they had a vesicle to excrete out into, you could imagine continually doing this, recovering them in the vesicles, and then later purifying them for spectroscopic or biophysical studies. So, I mean, is escort ever used for just membrane-associated proteins? You have just a cleavable leader sequence that's just being used to push your unstable protein into a vesicle.
Yes, I think I missed a question. I think it's a very exciting method to use with membrane proteins exactly because of that reason, because you will keep all your interacting proteins, and that I think they have a very important role in stabilizing a lot of these transmembrane proteins. But I don't know if you want to ask something in particular.
Well, escort is, I gather it's insect cell? It's insect cell machinery, but
No. This is mammalian cell machinery.
mammalian. Okay, all right. I mean, E. coli has like similar systems too. But yeah, I was thinking specifically about G proteins. We have some G proteins that are, really those are sort of membrane anchored or membrane associated. But we were trying with not escort, but a similar export system to try and get more of them in vesicles and then later purify them and work them up. And I'm just wondering if anyone's ever tried that with the escort system.
I'm not sure about it. I haven't seen G proteins with escort system. What I can tell you is you can also, if you don't want to have your escort attached to your C terminus, we also did try the puller system, so fusing the escort to the puller protein, so then your actual receptor would not have any extra amino acid that is not the endogenous. And it works as well. It just takes a little bit further, processing, because you have to get rid of the puller in the outside of the vesicle. But in our hands, there are no differences in the distribution of the protein on the vesicle, and the mass spec is as good as the original one.